The Principle of Biosafety Training by Stick Biosafety Committee, Meifa Luang University. This training program is intended to provide students and researchers with information necessary to comply with the requirements of the biosafety and biosecurity in Thailand. The information of biosafety and biosecurity base on the Pathogens and Animal Toxins Act, BE 2015, and Biosafety Guidelines for Modern Biotechnology, Biotech 2016. The objectives. You should be able to 1. Identify facility design, work practices, and PPE required for work at the BSL-1/BSL-2 level. 2. Employ standard precautions for BSL-2 agents while handling these materials. 3. Properly use a biological safety cabinet BSC. 4. Dealing with appropriate cleaning procedures for infectious material spills, BSL-2. 5. Understand and follow the purpose of the Pathogens and Animal Toxins Act, BE 2015. The laboratory environment can be a hazardous place to work. Laboratory workers are exposed to numerous potential hazards, including chemical, biological, physical, and radioactive hazards. For the biological hazards such as pathogenic microorganisms, pathogens such as bacteria, viruses, fungi, parasites, and prions that can cause diseases in humans and animals, toxins from living organisms, prions, recombinant DNA including viral vectors, gene therapy, cloning, diagnostic specimens, cultured of cells or tissues, stem cell from humans, animals, or plants. Others. Principle of Biosafety Control. To establish a control, regulatory, and supervisory procedure for researchers, workers, and prevent the unintentional release of biological materials into the environment, it is essential to consider both the infrastructure and procedures in accordance with the risk level associated with the research. At first, the researcher should do a risk assessment and then work under the containment. Factors for assessing the risk of pathogenic microorganisms. Biosafety and biosecurity are fundamental for protecting the laboratory workforce, the public, and the environment from exposure to and inadvertent or intentional release of harmful biological agents. A systematic approach to biosafety and biosecurity program management that reflects a four-element cycle: planning, assessment, implementation, and review and improvement should be implemented. For the Pathogens and Animal Toxins Act, BE 2058-2015, Department of Medical Sciences, Ministry of Public Health, the purpose to protect the public's health and safety against infections and animal toxins. 1. Regulation is covered on the operation on pathogens and animal toxins that affect to humans, animals, livestock, and beasts of burden, animals that are used as vehicles. 2. Pathogens that cause diseases solely in plants are legally invalid. Pathogens and Animal Toxins Act BE 2558-2015. Its function is to regulate, control, and prevent the usage of microbes and animal poisons rigidly in all government and private entities and to preserve the safety and welfare of the people. Section 16. No person may use pathogens or animal toxins for the following purposes. 1. To carry out any act which causes danger to other persons or other persons' health. 2. To carry out any act which causes damage to the environment. Section 17. No person may conceal, cover up, or destroy pathogens or animal toxins to obstruct a competent official's performance of duties in relation to disaster prevention and mitigation or provision of emergency medical service under the relevant laws. Section 18. For the purpose of pathogens control, the minister by and with the advice of the committee shall have the power to prescribe, in the notification, the list of pathogens, classified into 4 groups as follows: taking into account prevention methods, treatment methods, dispersal and quantities or amount of pathogens, in consistence with the degree of risk of causing diseases or hazards which may occur in humans, the community, livestock, beasts of burden, or other animals prescribed in the notifications issued under Section 6.3. 1. Group 1 pathogens, which include the pathogens that pose a low risk or low hazard. 2. Group 2 pathogens, which include the pathogens that pose a moderate risk or moderate hazard. 3. Group 3 pathogens, which include the pathogens that pose a high risk or high hazard. 4. Group 4 pathogens, which include the pathogens that pose a very high risk or very high hazard. Pathogens and Animal Toxins Act, BE 2558, 2015. Pathogen means 1. Microorganisms, 2. Biological substance, SARCU-FAP, 3. Other germs listed in the notifications issued under Section 6.3. Microorganisms equals parasites, protozoa, molds, yeast, bacteria, and viruses. Or 2. Biological substance means 2.1. A culture, any part or product of microorganisms or animal toxin, e.g. DNA, RNA, their toxin and protein. 2.2. Proteinaceous infectious particle, prion. Animal toxin means toxin from animals that causes a condition that prevents the body from working normally in humans, livestock, beasts of burden, or other animals prescribed in the notifications issued under Section 6.3. Pathogens and Animal Toxins Act, BE 2558, 2015. The regulated activities 1. Possess, 2. Produce, 3. Sell, 4. Import, export, transit. The pathogen list are classified into 4 groups based on prevention treatment methods, spreading quantities or amount of pathogens, and the degree of risk of causing diseases to humans, the community, livestock, beasts of burden, or other animals. Group 1, RG1, low risk, rarely cause infection in healthy hosts. Examples: specific laboratory strains E. coli, S. cerevisiae, P. pastoris, and some lactobacilli. Group 2 moderate risk agents may cause disease in a healthy host but are difficult to transmit, do not usually cause serious or life-threatening illness, and are readily treated or prevented. Examples: pathogenic E. coli, Shigella spp, Plasmodium spp. Group 3, RG3, high-risk agents usually cause serious disease, serious risk to laboratory workers, present significant community risk if spread in the environment, but there are usually effective measures for treatment and/or prevention. Examples: B. anthracis, hantavirus, yellow fever, HIV, cultures, SARS-CoV-2. Group 4, RG4. Very high-risk agents are those that present significant individual and community risks and usually produce life-threatening disease, are readily transmissible, and effective prevention and/or treatment is not usually available. Examples: Ebola, Hendra, and Nipah viruses. Animal toxin are classified into 3 groups based on treatment methods and quantities or amount of animal toxins, in consistence with the degree of risk of causing a condition that prevents the body from working normally, which may occur in humans, livestock, beasts of burden, or other animals. Animal toxin Group 1. Animal toxins cause a condition that prevents the body from working normally, but not at a serious level, and effective treatment is available. Group 2. Animal toxins cause a condition that prevents the body from working normally at a serious level, and effective treatment is available. Group 3. Animal toxins cause a condition that prevents the body from working normally at a serious level, and effective treatment is not available. Biosafety describes the containment principles, technologies, and practices that are implemented to prevent the unintentional exposure to pathogens and toxins or their accidental release. WHO, 2006. Other definitions of biosafety. The need to protect human health and the environment from the possible adverse effects of the products of modern biotechnology, i.e., the concept of biosafety as described in the introduction of the Cartagena Protocol, SCBD 2000. In terms of outer space, the concept of biosafety is referred to as planetary protection, the practice of protecting solar system bodies, i.e., planets, moons, comets, and asteroids from contamination by Earth life, and protecting Earth from possible life forms that may be returned from other solar system bodies. NASA, 2019. Biosafety objectives: protecting workers and the public against hazardous biological agents, protecting animal and plant health, dealing with uncertainty, protecting the environment. Biosecurity objectives: protection against loss, theft, misuse, diversion, or intentional release, preventing development of biological weapons and addressing bioterrorism. Addressing biosafety and biosecurity. Objectives in containment. While the objectives are clearly different, it is evident that biosafety and biosecurity are complementary disciplines that benefit from an aligned approach. It is therefore not surprising that biosafety and biosecurity in containment are often addressed together through a single biorisk management program, ensuring compliance with the requirements and good practices set out in both international guidance documents as well as in the different local legislative frameworks. Biosafety level? A specific combination of work practices, safety equipment, and facilities which are designed to minimize the exposure of workers and the environment to infectious agents. Agents assigned risk groups by WHO are RG1, unlikely to cause disease in humans or animals, low individual or community risk. RG2, may cause disease but typically not serious, individual risk, low community risk. Treatable RG3, may cause serious disease, usually treatable, high individual but low community risk, serious respiratory agents. RG4, serious or fatal, often not treatable, easy transmission, high individual and community risk. There are 4 biosafety levels depend on the agent risk group. Biosafety levels, BSL, different than the risk groups. Risk groups used in risk assessment. BSL are used in risk management. BSL are ways to control the agent facilities, safety equipment, practices, PPE, etc. Once risk is assessed, then the appropriate BSL is determined. Here is the notification of the Ministry of Public Health. RA. Characteristics of the place of production or possession of and operations on pathogens and animal toxins. BE 2063, 2020. Chapter 2. Operations Site. Level 1. Clause 7. Operation Site, Level 1 shall have the following characteristics. 1. Being a closed space or a sectioned room, whose inside is visible and which is sufficient for production or possession. 2. Having walls, floors, and ceilings designed and constructed using durable and easy-to-clean materials. 3. Having a door which is lockable. 4. Having equipment and tools sufficient for the quantity produced. 5. Having sturdy tables which can carry the weight of the quantity produced, have waterproof surface, can be easily cleaned, and are resistant to acids, alkalis, and disinfectants. 6. Having sturdy chairs made of non-absorbent and easy-to-clean materials with the appropriate size and sufficient quantity for operators. 7. Having hand-washing basins in areas of operations. 8. Having a space, an area, or a room for cleaning used equipment or materials. 9. Having a space, an area, or a room for collecting and storing solid waste by separating infectious waste from other types of solid waste, with measures to prevent access by unrelated persons, animals, and insects. 10. Having sound and temperature at a level that does not affect work and health of operators. And 11. Having adequate lighting for operations. The conditions in 4, 5, and 6 shall not apply to possession without production. Clause 8. Operation Site Level 1 used with animals in operations on pathogens shall have the same characteristics as specified in Clause 7 and the following additional characteristics. 1. Being an animal room separate from other rooms equipped with appropriate ventilation and temperature control. 2. Having a room floor made of materials that are easy to clean, smooth, strong, able to carry the weight, waterproof, and acid and alkali resistant. In case of floor drain, there shall be a tightly closed lid that can only be opened when draining water. 3. Having a self-closing door. And 4. Having characteristics of places, equipment, and animal care management which are in accordance with the laws on animals for science. Clause 9. Operational Site Level 1 shall have tools and equipment for operations on pathogens as follows. 1. Containers or packages of tightly closed containers which does not leak. 2. Tools and equipment suitable for storing containers. 3. Tools and equipment for transporting or moving which prevent containers from falling. 4. Waste bins with lids which can be opened without a hand touch. 5. Tools and equipment for destroying pathogens, such as an autoclave or other appropriate methods for destroying pathogens. 6. Equipment or saline solution for eye wash. 7. First aid kits. And 8. Biological spill kits with a minimum of liquid disinfectants, absorbent materials, and personal protective equipment, consisting of operating clothes, rubber gloves, safety glasses, and face masks, as well as equipment for collecting contaminated materials, such as tweezers, dustpan sets, and garbage bags for infectious waste. Clause 10. Containers or packages of containers specified in Clause 9 shall be labeled with information about scientific names and dates of production or containment. Clause 11. Operational Site Level 1 shall have at least the following safety and quality systems for operations on pathogens. 1. Having security of place of production or possession and controls of procedures on storage, production, import, export, sale, transit, or possession. 2. Having measures to control access by unrelated persons. 3. Using technically appropriate personal protective equipment. 4. Having collection, storage, moving, and disposal of infectious solid waste which are in accordance with the laws on public health. 5. Having technically appropriate cleaning of materials, equipment, and contaminated materials. 6. Recording transactions and quantities of productions or possessions. 7. Having Roper accident reporting and investigations. 8. Having practices for prevention and control of animals and insects in the operational sites. And 9. Having measures to prevent the spread of contamination to the environment. Biosafety Level 1 greater than good microbiological practice and of good occupational hygiene. 1. Restrict or limit access when working. 2. Door that can be kept closed when working. 3. Frequent hand washing. 4. Wear appropriate PPE, including disposable gloves and lab coat. Add eye protection if a splash hazard exists. 5. Prohibit drinking, eating, chewing, and smoking, handling contact lenses, applying cosmetics in the laboratory. 6. Pipetting by mouth strictly forbidden. Keep work areas uncluttered and clean. All surfaces, including floors, chairs, and bench tops, are easily cleanable. Decontamination of work surfaces after every use after any spills. No food in the lab refrigerator. Minimize splashes and aerosols. Maintain insect and rodent control program. Sharps precautions, including special containers for disposing of needles and other sharp objects. Change gloves when contaminated or the integrity is compromised. Never wash or reuse disposable gloves. Do not launder lab coats at home. Decontamination of laboratory wastes. BSL-2 laboratories are used to study moderate-risk agents that pose a danger if accidentally inhaled, swallowed, or exposed to the skin. agent associate with human disease. Facility construction requirements. Location, separated from public areas. Structure normal construction, ventilation, directional. Primary hazards. Accidental needle sticks. Exposure to eyes and nose, mucous membranes, ingestion of infectious materials. Agents do not cause lethal infections, are not transmissible via airborne route. Do not cause infection if tiny droplets become airborne and are inhaled, which might occur if the material were spattered. Agents are pathogens for which immunization or antibiotic treatment is available. Extreme care should be taken with contaminated needles and sharp lab instruments. Lab staff and students who work in BSL-2 labs must complete Introduction to Biosafety training policies to restrict access to lab containment during certain processes, i.e., aerosols, large volumes, etc. Biohazard warning signs posted outside lab, sharps precautions, hand-washing sinks, autoclave or waste decontamination, biological safety cabinet Class 2 BSCS desired. Use good microbiological practices, waste disposal, and aseptic techniques. Example, most non-respiratory, non-lethal agents. Standard microbiological practices. 1. Control access to the laboratory. 2. A documented safety manual must be available for all staff. 3. Personal protective equipment, PPE, to be used for laboratory procedures. PPE acts as a barrier to minimize the risk of exposure to aerosols, splash, and accidental inoculation. 4. Proper footwear. No sandals in lab. 5. Tie long hair back to prevent catching in the equipment or catching fire over Bunsen burner flames. 6. Eating, drinking, smoking, handling contact lenses, applying cosmetics, and storing food for human consumption must not be permitted in laboratory areas. 7. Mouth pipetting is prohibited. 8. Perform procedures to minimize splashes or aerosols, e.g., pipette carefully. Transfer biological solutions with caution. 9. Decontaminate work surface before leaving the lab. 10. Leave your lab coat in the lab. 11. Remove gloves before touching door handles, phones, etc. 12. Wash your hands with soap and water before leaving lab. 13. Safe storage of waste materials. 14. Decontaminate all cultures, stocks, and other potentially infectious materials before disposal using an effective method. A. Materials to be decontaminated outside of the immediate laboratory must be placed in a durable, leak-proof container and secured for transport. B. Materials to be removed from the facility for decontamination must be packed in accordance with applicable local, state, and national regulations. 15. When possible, use plastic instead of glass. 16. Safe handling of sharps such as needles, scalpels, pipettes, and broken glassware must be developed and implemented. Avoid manipulating needles after use. A. Needles must not be bent, sheared, broken, recapped, removed from disposable syringes, or otherwise manipulated by hand before disposal. B. Used disposable needles, syringes, glass pipette, and other contaminated sharps must be carefully placed in conveniently located puncture-resistant containers used for sharps disposal. Closable, puncture-resistant, leak-proof on sides and bottom, red/fluorescent orange container, or orange-red biohazard label. C. Non-disposable sharps must be placed in a hard-walled container for transport to a processing area for decontamination, preferably by autoclaving. D. Broken glassware must not be handled directly. Instead, it must be removed using a brush and dustpan, tongs, or forceps. E. Plasticware should be substituted for glassware whenever possible. This is Biosafety Level 2 room at STIC MFU. BSL-2 rooms shown Biosafety sign, a certificate of notification, contact person, and PPE for BSL-2. There are Biological Safety Cabinet Class 2 BSC, incubator, first aid set, biological spill kit, biohazard waste bins, eyes washer, lab sink, and hand washing sink. Biohazard Waste Disposal Diagram. Subordinate of Pathogens and Animal Toxins, Act BE 2558. Biosafety Manual Scientific and Technological Instruments Center, MFU. This manual applies to all workers, staff, students, visitors, and university employees who are involved in laboratory activities involving biological agents at the STIC building. This manual is in Thai. For foreign students and researchers, please refer to Biosafety and Microbiological and Biomedical Laboratories, BMBL 5th edition, and the WHO Laboratory Biosafety Manual, LBM. Detail in the manual. This is the Biosafety Management Procedures. Recommended National Biosafety Guidelines for Research Use of Microbial Agents, Pathogens, and Toxins. This is the Biosafety Guidelines for Modern Biotechnology. This is the National Laws and Regulation Pathogens and Animal Toxins Act, BE 2058-2015. Biosafety Level 3, BSL-3 laboratories are used to study agents that can be transmitted through the air and may cause potentially lethal infection. Researchers perform lab manipulations in Class 1 or 2, BSCs or other enclosure. Other safety features include clothing decontamination, sealed windows, double-door access, and specialized ventilation systems. Biosafety Level 4, BSL-4 laboratories are used to study agents that pose a high risk of life-threatening disease, aerosol-transmitted lab infections, or related agents whose risk is not known. Lab personnel are required to shower when exiting the facility. The labs incorporate all BSL-3 features and occupy safe, isolated zones within a larger building or a separate building. Procedures are performed in Class 3 BSCs or Class 2 while wearing a positive pressure full-body suit. The laboratory director is specifically and primarily responsible for assessing the risks and appropriately applying the recommended biosafety levels. Researchers are used to study biological agent, biological hazard, plant and animal or human pathogens, genetically modified organisms, recombinant and synthetic nucleic acids, animal toxins, biological toxins, human source material, non-human primate source material, samples from environment, insect vector. Requirements for all laboratories working with biohazards. They should be passed both of number 1 and number 2, and at least one form criteria, number 3 to number 5. 1. Biosafety and biosecurity training has been completed. 2. Laboratory equipment and safety are approved or trained. 3. Obtained at least 3 credits in a microbiology course. Or 4. Trained in good microbiology practice, techniques for performing necessary tasks, and ability to work safely, certified advisor. Or 5. By providing the operational training and techniques required to deal with infectious agents, certified advisors. Researchers' work involving the biological agents and biological hazard samples that pose low and moderate hazards to personnel and the environment. They should to submit their work to MFU IBC 02. They will be able to start working after approval. Students and researchers working with Agent Risk Group 2 are accommodated in the BSL-2 room. They should be properly trained for safety. 1. Biosafety or biosafety and biosecurity training. 2. Laboratory equipment and safety are approved or trained. 3. Standard microbiological practices, good microbiological practices, obtained at least 3 credits in a microbiology course. 4. Techniques required for the research work, special practices. Laboratory acquired infection, LAI. An infection obtained through laboratory or laboratory-related activities as a result of work with infectious biological agents, which may be either symptomatic or asymptomatic. Factors contributing to LAI 20% are caused by equipment failure, 80% of which are caused by human factors. Lack of knowledge of biological agent, lack of proper emergency responses, lack of training, passing down of bad habits, carelessness, ignorance in work practices. Risk assessment and management. The purpose of risk assessment and management are 1. Able to correctly identify and evaluate the risks associated with handling biological agents, and to put in place appropriate control measures to mitigate manage such risks. 2. To ensure that risks to employees, the public, or the environment are consistently minimized to an acceptable levels. Factors should be considered in a risk assessment for pathogen 1. Severity 2. Stability 3. Whether the pathogen is indigenous to the country. 4. Route of transmission. 5. Infectious dose. 6. Host range. 7. Availability of effective treatment preventive measure. To complete the biorisk assessment, 1. Agent characteristics and biological materials RGBSL, etc. include biological, chemical, radiological, liquid, nitrogen, flammable. 2. Personnel factor, personnel supervising and personnel using the material. Experience 3. Working activity factor. Experimental protocols and lab practices, SOP 4. Equipment factors 5. Environment factors, laboratory, facility, community. Risk assessment or evaluation, a process used to estimate risk against a given risk criteria to determine the risk. Risk equals severity times likelihood. Risk should be identified, evaluated, measured, and planned to minimize the risk. The most effective risk mitigation form: top to down 1, elimination 2, substitution 3, engineering controls 4, administrative controls 5, PPE. Who determines acceptable risk? Assessment is conducted by a biosafety professional in partnership with and based on information provided by the principal investigator. The assessment is presented to the Institutional Biosafety Committee, IBC, for approval. Working in a biological safety cabinet, you can find these video at Centers for Disease Control and Prevention, CDC's YouTube, or read the information that provided by National Institute of Health, Department of Medical Sciences, Ministry of Public Health. The usage and maintenance of biosafety cabinets in Thai version by the Department of Medical Sciences. The usage and maintenance of Biological Safety Cabinet by Scientific and Technological Instruments Center. Before using a BSC-1, check BSC positioning within room. 2. Check sticker for certification to ensure proper functioning. 3. Turn on blower and wait 5 minutes to clear air within the unit. 4. Decontaminate the sides, sash, and work surface by wiping them down with a suitable decontaminant. 5. Wear appropriate PPE. 6. Gloves should cover cuffs of lab coat. 7. Create a checklist of work materials that you will need to handle within the BSC. 8. Disinfect items as you load them into the BSC. Proper work procedure. 1. Work at least 4 inches from front grill. 2. Best work area, center 1/3 of BSC. 3. For greatest personal protection, work towards the back. Do not block the front grill. Proper work procedure 5. Work form clean to contaminated. 5.1 Set up BSC with a clean portion, a work area, and a contaminated, dirty area. 5.2 Do not move contaminated item over clean items. 6. Hands enter and exit BSC straight on. 7. Use slow, smooth movements. 8. Lateral motion may fan contaminants into or out of the cabinet. Do not block front grill. Block grill may provide contamination route. Do not block rear grill. Blocked rear grill disrupts airflow within BSC. Place materials in center for optimal performance. BSC safety. Never operate a BSC when it is alarming. Alarms may indicate the sash is too high, inadequate airflow, or the other operational issues. Only use the BSC with the sash at the appropriate height. Ultraviolet light safety 1. The efficacy of the use of UV light is debated. If bulbs aren't changed as required by the manufacturer, usually after about 4,000 hours of use, they may not generate intensity sufficient for effective decontamination. If BSC is periodically turned off and dust accumulates on UV bulb, it may not be as effective as necessary. UV light may not penetrate highly soiled surfaces or material, may create false sense of security that area has been decontaminated. 2. Proper use of chemical decontaminant may be more reliable. 3. CDC, NIH, NSF, and ABSA recommend against its use. 4. UV light is considered a probable human carcinogen. 5. Acute exposure effect, erythema, i.e. sunburn, and photokeratitis, UV damage to eyes. 6. Some BSC sashes to not fully close, increasing potential for exposure. 7. Risk presented by the hazard may not be warranted. Ultraviolet light safety exposure to UV light must be maintained below the occupational exposure limit. The limit for UV light, 6 millijoule per square centimeters. Cleaning spills within BSCs. Effective cleanup of spills within BSCs will help reduce the risk of contaminations. Hazardous chemical use. HEPA filters in BSCs do not remove hazardous vapors or gases. BSCs typically recirculate 80% of the air within the cabinet. May result in elevated concentration of toxic vapors or gases in certain types of BSCs. BSC exhausted outdoor ducted BSC, appropriate for limited use of hazardous chemicals at the work surface. BSC certifications. To ensure proper containment, BSCs must be certified, upon installation, annually, when moved, to a different room, when filters are changed, when repaired or modified. Segregating biohazard waste, please follow the chart. Personal protective equipment, PPE. Select the right type of PPE for each task at work according to risk assessment. Employer responsibilities to provide PPE and the employee's responsibility to wear it, maintain it, inspect it, and select the right type for the application. PPE will give the maximum protection when the person wear it, maintain it, inspect it, and choose the right type for the work. How to choose your PPE. 1. Perform risk assessment of the work. 2. Know which part need to be protected. 3. Know how much you need to protect that particular part. 4. Pathogen Safety Data Sheet, PSDS. Steps of removing PPE is very important as the PPE could well be contaminated with the pathogens. If the removing steps is not well managed, one can come into contact with the pathogen and the contamination can go far beyond the laboratory. Therefore, risk assessment should be performed and SOP should be in place. Sequences for putting on and removing PPE recommended by CDC. Guide for removing PPE. Point to considered and management of PPE. Medical surveillance. Exposure response. Incident reporting.